All right, let's jump right in. Today we're going on a pretty wild journey, deep into our own DNA, and billions of years into the past, all to answer a question that sounds like it's straight out of science fiction. We're going to look at the instruction manual for life itself and ask, can it actually get full?
I mean, really think about that for a second. Our genes are the blueprint for literally everything about us. But can we just keep piling on more instructions, more features, more complexity forever? Or is there a breaking point where the whole system just gets overloaded?
So to even start to answer this, we've got to talk about something called gene regulation. Here's a simple way to think about it. If your genes are the computer hardware, then gene regulation is the incredibly complex software that runs the whole show. It's this system of switches and dials and knobs that tells every single gene when to turn on, how loud to be, and when to shut off. It's the reason a brain cell is totally different from a skin cell, even though they have the exact same DNA.
And logically, you'd think that software can't just get infinitely complex, right? I mean, imagine a single gene with, say, 200,000 different settings. The amount of code needed to control that would be insane. And the chances for bugs, or in this case, harmful mutations and diseases, would just go through the roof. So there has to be a ceiling somewhere, doesn't there? To figure this out, a team of scientists came up with this really brilliant approach. Instead of trying to guess what happens in the future, they decided to look into the past. They asked a simple question: what happens to the complexity of a gene as it gets older? Does it get more regulated, or maybe less?
And we're talking about a mind-boggling amount of time here. The study sorted human genes into groups based on how old they are. So the youngest ones are just found in primates, showing up less than 80 million years ago. But the oldest genes, whoa, they're ancient relics dating back over 1.6 billion years to the very first complex cells on this planet. That's the ancestor of every plant, animal, and fungus today.
Okay, so with this game plan in mind, the researchers laid out 4 possible scenarios for what they might find. 4 potential futures, you could say, for the relationship between a gene's age and just how complex its regulation gets.
All right, so this slide shows you the 4 main possibilities. You can think of these 4 graphs as the potential answers to our big question. Does complexity just keep climbing up and up forever? Does it eventually hit a wall and just stop? Or is something totally different going on? Let's walk through each one. First up is scenario A. This one's the most straightforward idea. As genes get older, they just keep collecting more and more regulatory controls. It's a straight, upward sloping line with no end in sight. It's like our genetic software just keeps getting more and more features with every update, forever.
Then you've got scenario B, which is called saturation. In this version, complexity goes up for a while, but then the gene just gets full. It hits a maximum capacity, a ceiling, and the line just flattens out. You just can't cram any more instructions in there without breaking the whole system.
Now, scenario C suggests a totally different strategy. Imagine it's not about just adding more rules, but about swapping old rules out for new ones. So maybe evolution uses one style of regulation for young genes and then a completely different style for the ancient ones. The total amount of complexity might stay about the same, but the type of complexity changes over time.
And finally, there's scenario D. This is basically the "what if there's nothing here" option. It's the null hypothesis, that there's just no pattern at all. A gene's age and how complex its regulation is are totally separate things. It's all just random.
So, those were the 4 possibilities on the table. To find the real answer, the scientists had to get their hands dirty and actually go in and measure this complexity across thousands and thousands of genes. And let me tell you, they were incredibly thorough. They didn't just look at one thing, they measured complexity in 8 different ways. For instance, they counted the number of transcription factors. You can think of those as the little proteins that act like on-off switches. They looked at alternative splicing, which is this cool trick that lets a single gene create multiple different protein recipes. They even tracked these tiny molecules called microRNAs that act like volume dials, fine-tuning a gene's activity. The point is, they looked at this from every possible angle.
And the results? Well, you can see them for yourself. They were stunningly clear. You don't have to worry about the details of each of these charts. Just look at the general shape. In almost every single one, the line goes up and to the right. What that means is, as genes get older, that's moving from left to right on the bottom, their complexity just consistently goes up. And this table really puts some hard numbers on it. I mean, look at this. The oldest genes, on average, produce more than twice as many protein variations, or isoforms, as the youngest genes. It's the same deal for the number of start sites that kick off a gene's activity.
The big takeaway here is that this isn't some small trend. It's a huge, undeniable increase in complexity.
But the number that is just absolutely wild is this one. When they looked at those tiny microRNA targets, the oldest genes have, on average, over 33 times more than the youngest genes. 33 times! That is just a staggering amount of complexity being added over evolutionary time.
And remember, this wasn't just true for one or two things they measured. For all 8 different types of regulation they looked at, the story was exactly the same. The data was practically screaming in one unified direction. Older is more complex.
So let's circle all the way back to our 4 original scenarios. Saturation, the idea that genes get full? Nope. The lines just kept on climbing. A trade-off, where old rules get swapped for new ones? Nuh-uh. Complexity just went up across the board. No connection at all? Clearly not. Only one single hypothesis fit all the evidence perfectly. Scenario A, a continuous, seemingly unending increase in complexity.
So what does this all mean? Well, it paints this incredible picture of our genome not as some finished product, but as an ever-evolving, ever more intricate masterpiece that is very much still being written.
The core takeaway is this: our genes have not hit their limit. There is zero evidence that they are full. This process of adding new layers of control, new switches, new dials, a process that has been going on for over 1 billion years, is still happening right now.
And that leaves us with this truly profound final thought, which comes straight from the researchers themselves. Our genetic code, as unbelievably complex as it already is, apparently has the capacity to become even more complex. Evolution, it seems, is nowhere near finished with us. You have to wonder, what new layers of control will our descendants' genes have 1 million years from now? The code is still being written.